Today, we're jumping into something that sounds like it's straight out of a sci-fi movie, but has very, very real consequences for all of us: your genetic data. It holds the keys to unlocking personalized medicine, but only, and this is a big only, if we can actually read it correctly.
So imagine this. You send your DNA to two different top-of-the-line labs. You'd expect to get back the exact same results, right? I mean, it's the same DNA, but what if I told you that, depending on which lab you sent it to, you could get two completely different medical diagnoses? It sounds crazy, but that is the high-stakes reality we are facing right now.
And look, this isn't some minor statistical fluke. When different labs analyze the very same genetic data, the results can be, well, shockingly inconsistent. This isn't a theoretical risk. It's a gamble, a real gamble with patient health. And honestly, the odds aren't great.
Get ready for this number, because it's pretty staggering. For the most common type of genetic variation, we're talking a single-letter change, like a tiny typo in the book of your life. Different labs only agree about 60% of the time. That means there's a 40% chance of disagreement. That gap could be the difference between a doctor spotting a critical gene for a disease or missing it entirely. But believe it or not, it actually gets worse.
For variations that are just a little more complex, like bits of genetic code being added or deleted, the consistency rate just nosedives to less than 30%. I mean, stop and think about that for a second. More than 2/3 of the time, the results from Lab A will not match the results from Lab B. It's a huge problem.
So here's where the rubber really meets the road. One study found that over 16% of variations that have direct clinical importance, the ones that really matter for a diagnosis, were only caught by one specific method. That means another lab, using a slightly different method, could completely overlook a mutation that is absolutely critical for figuring out what's wrong or how to treat it.
Okay, so why? Why on earth is this happening? Well, the answer lies inside something called a bioinformatics pipeline. It's basically the digital process that turns all that raw DNA goo into a medical report. And it's the exact place where all these dangerous inconsistencies are born.
You know, the easiest way to think of a pipeline is like a digital recipe, or maybe an assembly line. It takes this messy, raw data that comes spitting out of a DNA sequencing machine, and step by step, it cleans it up, organizes it, and refines it until you have a neat, clean list of genetic variants a doctor can actually understand.
So this assembly line has three main stages. First up is quality control. This is like washing your vegetables. It just cleans up all the errors and the digital noise from the machine. Second is sequence alignment. This is where every little piece of your DNA is mapped to its correct spot on a standard human genome map. And finally, you have variant detection. That's where the computer compares your DNA to the reference map to spot all the little differences that make you, well, you.
And here it is. This is the root cause of the entire problem. Each lab is basically using its own custom recipe. They might use different software tools or different versions of those tools, or even just tweak the settings a tiny bit at each step. It's like telling a dozen chefs to bake the same cake, but they all use slightly different ingredients and oven temperatures. You're going to get a dozen different cakes.
So that brings us to the pretty obvious solution, right? Standardization. If every single lab followed the exact same validated recipe, we could wipe out this dangerous inconsistency. And this isn't just some nice idea. It's an absolute clinical necessity.
There are really three huge reasons this is so critical. First, and obviously the most important, is patient safety. We need results that are accurate and repeatable, every single time, no exceptions. Second, it's about collaboration. If everyone's data can speak the same language, we can pool it together for massive research studies to cure diseases. And third, it's about regulations. Clinical labs have to meet strict quality standards, like ISO 15189, to prove their tests are reliable.
The great news here is that we actually have the tools to do this. This isn't a pipe dream. So let's take a look at the toolkit that makes this universal standard possible.
First up is something called a Workflow Management System, or a WMS. Just think of it as a digital master checklist. It automates the whole pipeline, and it forces every single step to run in the exact same order with the exact same settings. It just takes human error right out of the equation. Tools like Nextflow are making this a reality in labs today.
The second key piece of the puzzle is containerization, and the analogy for this is brilliant. The digital lunchbox. A container made by software like Docker packages up a tool and its entire environment, all the little dependencies it needs to run, into one neat little box. That means the tool runs exactly the same way on a computer in New York as it does on a server in Tokyo. It's identical.
And this just lays out the difference perfectly. It's night and day.
The old way was a mess. It was manual, inconsistent, full of human error, and a nightmare to trace. The new way, using this toolkit, is automated, standardized, totally repeatable, and completely traceable. It's a genuine revolution in reliability for this field.
So what does this all actually mean for doctors and, more importantly, for patients? Well, the impact is huge. It means getting diagnoses faster, finding treatments that actually work, and ultimately better outcomes for people.
And this isn't just a theory, okay? Listen to this quote from the research. By standardizing and automating their process, labs have literally cut down the reporting time from 10 days to 7. Now, for a patient anxiously waiting for those results, those 3 days, well, they're everything.
Let's make this even more real. Take the case of a lung cancer patient. A standardized report came back and it clearly, reliably pointed to one specific mutation, EGFR L858R. This wasn't some ambiguous guess. It was a crystal clear signal that let doctors immediately pick a specific targeted therapy known to work best against that exact mutation. That, right there, is what precision medicine is all about.
Now you can see from this image, the world of genomics is just mind-bogglingly complex. There are countless pathways and data points, but the core message here today is simple. Standardization brings order to all of this chaos. It turns a ridiculously complex map into a reliable guide that doctors can trust. And the story doesn't even stop there. The future is getting even more exciting. The next step is to make these standardized pipelines not just automated, but truly intelligent. AI tools like Google's Deep Variant are already learning to spot mutations with superhuman accuracy, which promises to make these digital recipes even more powerful. Which leaves us with one final massive question. Standardization is the critical first step, but if we can get every lab all around the world producing data that's consistent and comparable, could we build a global library of human genetics? A resource so vast that it could help us finally crack some of medicine's oldest and biggest mysteries? The technology is here. The only thing left to ask is if we can all work together to build it.